Good morning. Hi.
How's it going, Eric?
I'm all right.
Very good. We're going to go through kind of a lot about how the spinal cord is organized today, so we can kind of get a better understanding of exactly where the information that we're receiving from our body, which part of the spinal cord that information is carried up on. And then also when we give our body motor commands, where in the spinal cord that's occurring. So when we went over the brain, we talked about the different tracks. We had our commissural tracts that was going through the corpus callosum, basically allowing right to left side brain communication. We had our association tracts, and those were more kind of front to back between the different lobes of the brain or even within lobes of the brain. And then we had our projection tracts. So the projection tracts are the ones that are basically just groups of axons that are going to be carrying either motor messages down from the brain through the spinal cord or sensory messages coming up from the body through the spinal cord. So most of this is really about those projection tracts and kind of how that information is being carried through the spinal cord to help the body organize itself. My computer's doing some updating stuff. So let me share my screen here. And get back to where I can see all of you. And whoops, I don't think I shared the correct screen. I clicked some other boxes to make the audio and stuff work better, but then I forgot to click which screen I wanted to share. So that didn't work. Now I'm good, I think. And pull up my chat so I can pay attention to that. And my participants. And pull that over. All right, I got too many things open, too many browsers. All right, now I'm good, I think. So, this chapter kind of refers to those tracks as pathways, and it's really kind of a synonymous term. Essentially, it's just this grouping of axons that have common functionality that are lumped together. And it has significance when we have people that have like spinal cord injuries or spinal cord tumors. If you're getting spinal cord compression from a tumor in a certain area of the spinal cord, it may not affect the entire function at that level of the spinal cord. It might only impair the area where that tumor is bulging in and compressing. So sometimes people will start having some weird symptoms, and that can be an early sign that they've got something going on that's causing some spinal cord compression, whether it's some kind of bony type narrowing, like from arthritic type conditions internally, or whether it's more like, say, of a lesion, a mass that's growing in the spinal cord and taking up space and basically causing some compression elsewhere. So we're going to start out with a little video that kind of goes through the whole big picture. It's probably a little bit more detailed than I expect you to know in certain areas, but I thought they did a pretty good job of kind of giving a good overview of how things are organized. And then we'll kind of work our way through the chapter after the video and kind of highlight the things that they talked about in there. You'll find that when I play videos, I usually have either a video choice that's going to be somewhat amusing, hopefully to you, or it's somebody with a foreign accent, because I think it's easier to, just more interesting when you're having to concentrate a little bit harder on what these people are saying. So I'm not sure if this person is from Australia or England or where, but it's kind of an English type accent. I probably have to listen to it again to let you know. So hopefully it'll all play for me well, and we'll see. It's about 8 or 10 minutes long, I believe. And it's taking a while to load. Okay, let's see. It did this to me yesterday. It decided it wasn't going to play, and then I had to do it again a different way. Well, I thought I was going to play it. 51 minutes, that's not what I want. Well, this may not work, unfortunately. If it doesn't, it doesn't. Let me try one more thing. See if I can go back into it. Let it refresh. And maybe it'll be happy for me. Apparently not. I played it yesterday, and the first time I tried it, it gave me this message, but the second time it went through, so I don't know why it's not today. Probably because I'm trying to hold class. So apparently we'll just, nope, not here. Have mercy on me. You guys hear it okay?
Okay, so in this part of the tutorial, what I'd like to do is introduce you to the idea of the different ascending and descending spinal pathways. So hopefully you will have watched the previous parts on the spinal pathways and will understand that the ascending tracts carry sensory information from the periphery to the brain, and the descending tracts carry information from the brain to the periphery. So what we're looking at here is a horizontal section of the cervical spinal cord, and you'll recognize the H-shaped grey matter in the middle, and around it is the white matter. And there's various different shapes that you'll see here, and these represent the different ascending and descending spinal pathways. So first we'll take a look at the ascending pathways. So we'll look first at the dorsal columns, which convey fine touch, proprioception and vibration sense from the periphery up to the brain. So you've got the fasciculus gracilis, which I've highlighted in yellow, and you've got the fasciculus cuneatus. So just remember that fasciculus means small bundle. So the fasciculus gracilis carries information from the lower limb, so it has axon fibers from the lower limb, whereas the fasciculus cuneatus has fibers from the upper limb. So the way I remember this is that the letter C is before the letter G in the alphabet, so C is higher and your upper limbs are higher than your lower limbs, so fasciculus cuneatus carries fibers from the upper limb, whereas fasciculus gracilis carries fibers from the lower limb. Now how do you remember which one is medial and which one is lateral? So in yellow we've got the fasciculus gracilis, and in green we've got the fasciculus cuneatus. And how to remember this is that your arms are lateral to your legs, and you know from the previous mnemonic that the fasciculus cuneatus carries fibers from the upper limb. So because your arms are lateral to your legs, the fasciculus cuneatus is lateral to the fasciculus gracilis in the spinal cord. So next we've got the spinothalamic tracts. So these I've just highlighted in blue. And the spinothalamic tracts are also known as the anterolateral tracts, as they sit anteriorly and laterally in the spinal cord. And some texts will refer to distinct anterior and distinct lateral tracts, but almost invariably there is some intermingling of fibers between the anterior and lateral tracts. So on this diagram they're represented as one tract. So the anterolateral tract is known as the spinothalamic tract, and this relays information to the somatosensory cortex regarding pain and temperature information and also crude touch. So the lateral aspect of the spinothalamic tract carries pain and temperature, whereas the anterior spinothalamic tract fibers carry crude touch information. So the next ascending pathway is that of the spinocerebellar pathways. So I've highlighted these both in purple and lighter purple. So you've got the dorsal and ventral spinocerebellar tracts. So these tracts relay information from muscle and joint receptors to the cerebellum, and these tracts are examples of the pathways which carry subconsciously processed information, whereas the dorsal columns, the fasciculus gracilis and the fasciculus cuneatus, and the spinothalamic tracts carry consciously processed information which is processed in the somatosensory cortex. So these spinocerebellar tracts don't carry information that is consciously processed, so they don't have the 3 neuron sequence which I described in the previous part of the tutorial. So basically you've got information coming via these tracts from muscle spindles and Golgi tendon organs, and this information is relayed to the cerebellum and allows for the control of posture and to coordinate muscular movements. So now we'll take a look at the descending tracts. So these descending tracts start in the cerebral cortex and brainstem and descend down the spinal cord to innervate muscles. So they are involved in controlling movement, muscle tone, spinal reflexes, and also have an ability to modulate some of the sensory information which is brought up via the ascending tracts. So essentially the ascending tracts bring in information from the environment relating to sensory information, and the descending tracts carry information from the center, the brain, to act in response to this sensory information. So it's useful to think of these descending tracts as pyramidal or extrapyramidal, and pyramidal just refers to tracts which pass through the medullary pyramids. So the pyramidal tracts are the corticospinal tracts. So you've got the lateral corticospinal tract and the anterior corticospinal tract, which I've highlighted in orange. So the corticospinal tracts are important to know about because they convey voluntary, skilled movements originating from the motor cortices of the brain. So the lateral corticospinal tracts carry motor information destined for the limbs, and it's important in skilled voluntary movement. The ventral corticospinal tract carry axial motor information. So the axial muscles are those central muscles which aren't limb muscles essentially. So you may start to notice the significance of the naming of these tracts. So remember the descending tracts we talked about, the spinothalamic tract and the spinocerebellar tracts. You'll notice that the first part of the name of the tract relates to the first where the tract begins, and the second part of the name of the tract relates to where that tract ends. So spinocerebellar, it starts in the spinal cord and it projects the cerebellum, and then spinothalamic starts in the spinal cord, projects to the thalamus. So you know that they are ascending tracts. And now we've just talked about the corticospinal tract, so starts in the motor cortex and projects down into the spinal cord. So it's a descending tract. So there's always clues and hints in the names. So just to cover some of the extrapyramidal tracts, you've got the rubrospinal tract, which I've highlighted in red. So this originates in the red nucleus of the midbrain tegmentum and is responsible for limb flexors. And then you've got the tectospinal tract, which I've highlighted in light purple here. So it's seen here kind of intermingled with the medial longitudinal fasciculus. But the tectospinal tract originates from the superior colliculus of the midbrain. So the clue's in the name again. So the tecto part of this word refers to the tectum, which consists of the superior colliculus and the inferior colliculus. So fibres from the tectospinal tract mainly terminate in the cervical segment of the spinal cord and carry information responsible for movements in response to visual sensory information. And then we've got the vestibulospinal tract. So this is highlighted in bright pink, and this originates in the vestibular nuclei in the pons and the medulla. So you've got a lateral vestibulospinal tract and a medial vestibulospinal tract. So highlighted here is in pink is the lateral vestibulospinal tract. So the vestibulospinal tract receives information from the bony labyrinths and the cerebellum relating to balance and posture, and this pathway allows for the control of extensor motor neurons in order to maintain posture. And then lastly we've got the reticulospinal tract, which arises, as the name suggests, from the reticular formation of both the pons and the medulla. So you've got a medial or ponto-reticulospinal tract, which is the one highlighted here in green. And then you also have the medullary reticulospinal tract, which aren't shown here, but I'll just draw them on the rough location of these fibres. So you'd have the medullary reticulospinal tract fibres in this region here. So that's the lateral reticulospinal tract. And this tract, the reticulospinal tract, is responsible for reflexes, muscle tone, and is also involved with the respiratory and circulatory system. So that's an overview of the different ascending and descending spinal cord tracts.
All right. Let's see, let me close that down. Yeah, so I saw Tom's message about that. I guess this isn't shown in the link that's in Canvas for the set of slides, but if you are interested in possibly watching it again, it is Anatomy Zone, is the overall series that kind of goes through all these. And so they are pretty well done. Again, this one goes into a little bit more detail than I expect you guys to know on some of this, but we'll kind of go through the slides today, and I'll try and emphasize the more important stuff to kind of link together. So hopefully a lot of the terms and kind of discussion that the guy went over, talking about these different parts of the brain and how these different pathways lead from one area to another, didn't overwhelm you too much. But we'll kind of, like I say, touch on all the key areas and make sure that it's pretty clear for everybody before we move off of this topic. All right, so ascending, anytime you hear ascending, you should be thinking sensory. That's information coming from the body to the brain. Anytime you hear descending, it's going to be some kind of motor function, whether that's physical movement or whether it's like gland secretion or smooth muscle contraction, things like that. So ascending and descending. And again, we've got those kind of similar terms, afferent and efferent. And so the afferent is going to be the ascending and the efferent is going to be the motor. So all those terms are basically synonymous to one another. All right. So he mentioned briefly, when we looked at those descending paths, we had the pyramidal tracts and then the extrapyramidal tracts. So the pyramidal tracts are going to be the ones that specifically are going to decussate at the medulla oblongata in the pyramids. If they're extrapyramidal, that means they travel somewhere else in the spinal cord where they're not necessarily going to be crossing over at that level. Either they're not crossing over or they cross over someplace else. But most of the things are going to cross over. And we've kind of talked about that. We've talked about this concept of somatotopy. And that's, we didn't use that term, but that's basically when we looked at the sensory and the motor homunculus. When we looked at the precentral gyrus and the postcentral gyrus, those specific areas that we noted where the foot, the ankle, the hips, the abdomen are controlled, or we receive sensory information and they can kind of map that out along those two gyruses, the precentral and the postcentral gyrus, whether we're talking about either the motor part or the sensory part. So that's all that big word somatotopia refers to. All right, so our sensory pathways, which is what he started the video with, those are our ascending pathways. Different areas of the spinal cord are going to send our brain different type of sensory information. So we've got both somatosensory, the things that we are consciously aware of, and then viscerosensory, the things that our body needs to be aware of, but we may not be aware of. Those internal organ sensations, chemical changes, sensations, things like that are going to be in the viscerosensory. All right, so when we look at our sensory pathways, we're typically going to have either 2 or 3 neurons involved in the chain. The 3 neuron is the more common one. The 2 neuron chain is really mostly seen with information coming into the cerebellum because it doesn't have to go to the thalamus first. The things that we need to be aware of, the somatosensory stuff, it's going to have an additional synapse essentially at the thalamus, and then the thalamus is going to have another axon that goes out to the particular part of the postcentral gyrus that is, receiving that information from right here on my arm or from my face or whatever the case might be. So we've got our primary neuron is going to be the one that has the sensory receptor to it. The cell body for that is going to be in that posterior root ganglia, which we've talked about. And then that is going to travel up through the spinal cord and then typically synapse somewhere either in the spinal cord. Brainstem is the most common location. and then it's going to ultimately connect to the thalamus, and then the thalamus is going to send its axon to the actual part of the cerebral cortex in that postcentral gyrus. So that's kind of the 3 neurons that we typically see. Again, if the information is proprioceptive information that's just going to the cerebellum, then it doesn't go up to that higher level directly. The cerebellum will then influence kind of the motor activity through its actions, but the actual sensory receptor neuron for proprioception, those things that we went over last time, the Golgi tendon organ, the muscle spindle, that's just going to synapse into the cerebellum, and the cerebellum is going to make sense and figure out how it needs to use that information to help the body function a little better. So the terms up here are a little bit different than what the guy in the video talked about. So he did mention the dorsal column. So dorsal is back. Think about dorsal fin. So those are the most posterior columns that we looked at, kind of in the middle of that H when we look at the gray matter. If I hold up my little model right here, this is the front, this is the back. So those dorsal columns are going to be these areas here. And so that's that fasciculus cuneatus and the fasciculus gracilis that are found in that, what's this term is calling, this book is calling the posterior funiculus. The medial lemniscus refers to that same pathway, but just in a higher portion of the brain. I think it's more kind of in like the brain stem is then what they call that. Think of it kind of like a road that changes names when it crosses a certain boundary. It's the same pathway, it just has a little bit different name. And then the anterolateral pathways, he mentioned as the spinothalamic. And again, these names can tell you a lot. If it's spinothalamic, it means that information is going from the spinal cord to the thalamus. So that's ascending. It's going from some more distal location to a more inferior location to a more superior location with that. And then our spinocerebellar, that's going to be that 2 neuron chain that's just going from the spinal cord and then synapsing at the cerebellum itself. So this image should look kind of familiar, but pretty much like he had in his video there. And so we've got in the back in those dorsal columns, the gracilis is the more central pair. We've got a right and a left half, and these are going to give us sensory information coming from the legs. And again, for me, what I kept straight, gracilis, that's one of our leg muscles. So that should be a big hint that that's the leg one. The cuneatus is the more lateral pair within that dorsal column, and that's going to be sensation in the arms, essentially, when we look at that. Our spinocerebellar tracts are these more lateral located tracts, and then the anterolateral pathways are going to be these ones that are more towards the front. You can see on this diagram, this light blue coloration, we've got this slide kind of separates those 2 areas more distinctly. The guy in the video kind of lumped them together and kind of had a bigger arcing kind of color for that area. But this text chooses to kind of say this is the lateral spinothalamic tract, and then we have the more anterior one. And they do carry specific information for different body parts. So we'll go through each of those real quick. So when we look at those dorsal columns, the 2 fasciculus ones, what we're receiving from that information is a little bit on limb position. So if I close my eyes and I bend my elbow, I'm receiving information that's telling me what kind of bend I have at that elbow. And now if I'm working accurately, I can also match myself on the other side. If for some reason that information wasn't being received correctly, I might not be able to match that same angle with my arm. But it is that internal sense of where is my arm that helps us then replicate that position on the other side. And again, we're going to have some commissural tracts going on to help us do that too. So that's kind of an example of that joint position sense or limb position. We also have discriminating touch. So really fine touch. So if you imagine like you have a little spider crawling across your arm or some little bug crawling across your arm, it's not much weight. It's not really very forceful, but we still feel it. That's kind of our fine motor touch, fine discrimination, as opposed to you put a tight belt on and cinch it up really tight, you might feel that belt, but that's not fine touch. That's more of a pressure sensation that you're feeling with something like that. So we do have precise pressure also. You're gonna have both deep pressure and precise pressure. Precise pressure, think about like maybe you had that little bug crawling on you and you're a very sympathetic, empathetic person and you don't want to hurt that little bug and just like smash it. So you want to very delicately pick it up and take it out, carry it outside and release it to be free. You need to have really good precise pressure so that you don't just pick it up and crush it. And so like little kids, when they're learning how to control their bodies, that's not really fine tuned. And so sometimes they will mash stuff, break stuff because they just grip it too hard. And when people lose a limb, like if they lose their hand and have an amputation and they get an artificial hand, that's been one of the hardest things for people that build those components to master, is helping figure out how we can make that hand grab something, but how do we know how to give it information about how tightly they need to grab it. And so if you're picking up, let's say you're picking up a plastic water bottle, just those disposable throwaway ones like a Dasani bottle or one of the more flexible ones. If you grip that thing too tightly and it's so flexible, you're just going to crush it and the water's going to spill everywhere. So you kind of need to be able to sense through that discriminating touch and this precise pressure how to kind of control that. So that type of information is being carried in that fasciculus cuneatus. And then we also get vibration. Hang on a sec, I got to let my dog in. I hear her scratching. I'll be right back.
So here's a question.
Uh-huh.
So if you had like some sort of weird magical wooden hand, it wouldn't work that well unless it has sensory things because it could grip stuff too tightly and then it squish stuff.
Yep.
Or it just can't hold on to it that well.
Right, because if you can't get that sensory feedback in, you don't know how much force to generate to grasp whatever you're trying to grasp.
So a wooden hand wouldn't be that useful even if it was animated, unless it had sensory organs.
Right, or you had some limits on how much force it could generate, that you might be able to control things that way. So all of that same information is going to be carried for the legs in fasciculus gracilis. Fasciculus cuneatus is the arm. So we probably have more neurons dedicated to that stuff in our arms. We tend to have better sensitivity in our arms and in particular in our hands because that's what we manipulate objects with. And so we need a lot better ability to identify what we're holding, how much force we need, and those type of things.
Here's another question.
Uh-huh.
Is there senses in the body to like know when to let go, or like say if you grip on something so tight it's gonna actually injure your hand? Like say you have a wooden hand.
Yeah, I mean, let's say you're picking up something sharp, you drop a glass and you're picking up the pieces of glass, then you would definitely, if you grip too tight, you're going to get hurt and you're going to receive that information of pain through those nociceptors. And that's kind of how we learn our lessons sometimes, is the hard way.
Well, I was thinking like holding on something tight, you actually hurt some parts of the hand, even if it was a wooden one, like some structural parts.
Yeah, I mean, you could grip so hard that you could break whatever is gripping with it potentially, or make it not function quite as well. You might impair its function a little bit if you damage the gear or some other mechanism kind of within it. All right, so we've got our anterolateral pathways. Does somebody else?
I just had another quick question. So those are the specific things that those receptors pick up, but those ones don't pick up pain.
Correct.
There was that other receptor I was talking about that picks up pain, right?
Yes. Uh-huh.
Okay.
Yep. So our anterolateral pathways, the ones that are going to be more if we look at our spinal cord model again, that's going to be the stuff that's kind of more in this region, anterolateral. This is our anterior horn. This is our lateral horn. So anterolateral is going to be kind of in here. So if we look at the anterospinothalamic pathway, we're going to get crude touch and pressure. So that's more like if I cinch that belt up really tight, then that's going to be carried on that pathway, where I just have a lot of pressure. Or if I'm, crude touch is more just, I know something's touching me, but I don't recognize exactly what it is. Discriminating touch is more that you can identify what it is. We'll do a couple different things related to discriminating touch. One is what's called 2-point discrimination. And that's basically, we have a device that has like 2 prongs to it. And when you put the prongs close enough together and you touch down on a body part, it'll just feel like one, because the same sensory receptors is picking up that information and it can't really tell that it's 2 distinct things touching you. And so we'll gradually kind of move those points further apart and touch it down, move those things further apart and touch it down. And at some point, the person will recognize, oh yeah, now I feel 2 things touching me. Well, 2 things were touching him the whole time, but we couldn't discriminate that. So that's kind of an example of discriminating touch. Another example would be a thing called stereognosis. And so stereognosis is the ability to recognize objects by touch. So let's say you reach into your backpack, you reach into your purse or whatever, and you're reaching around to find that pen and you're like, oh, there it is. You can pull out your pen. That's stereognosis. If we didn't have that discriminating touch, we wouldn't be able to recognize the shape, the texture of that pen or that safety pin or that button or that quarter. That's the ability that gives us the ability to recognize the difference between a quarter and a nickel and a penny and a dime and things like that. So crude touch is much more general. You're like, you reach your hand in and you're like, oh yeah, I feel something in that bag. I'm not sure what it is. So that's kind of the more crude or gross kind of touch. Our lateral spinothalamic tract, that's where we're going to get pain. So that's where the nociceptor information is going to be carried. That's where our thermoreceptor information is going to be carried. So when you touch that hot stove or pot handle that you've been cooking with, that sensory information is not being carried on that fasciculus section, fasciculus cuneatus. That's going to be in this lateral spinothalamic tract. And when we test people's sensation, generally we want to clear both of those areas. So if you can identify that they can accurately pick up either pain or temperature, you can be pretty sure that both of those functions are still working. If you can make sure that that person can recognize discrete fine touch, then you kind of know that you're covering light pressure as well as fine touch and proprioception and vibration sense. So as long as we clear one of the different things that is carried in those pathways, then we know that that tract is still working and it's not impaired. So those are
about that, Craig. So when you were talking about light touch, I was actually going to ask about the pressure one as well. When it comes to, like, let's say arm wrestling, why is it that we're more apt to not like break the other person's hand and we're more apt to pull a bicep? Are we just like not squeezing? Because I know we always squeeze whenever you grab because you're trying to pull the other hand down, but you think you'd be more apt to break the other person's hand.
Yeah, I mean, I think you're trying to match that person's grip so that you stay in contact with their hand. But really, the action that we're trying to do is kind of that internal rotation of the shoulder, essentially to pin that person down. So it's really, we're maintaining a certain amount of force at the hand, but the goal isn't to like squeeze their metacarpals together. The goal is. So
is that kind of like light touch? Like we're not really focused on trying to squeeze to the death out of it, because we know we're able to break a hand.
I don't know that it's necessarily light touch. It would be more probably kind of pressure. And it would probably almost be more the deep pressure part, because it's not like you're just barely touching hands. You're definitely making a firm grip. And the more firm that contact is, then the more that deeper signal will kind of take over and we'll lose some of the light touch sensitivity. Because it's getting masked essentially or overridden by the stronger signal of, oh yeah, I'm squeezing together. So the more force that's being generated, the more often that signal is going to get sent, as opposed to like a fly lands on me and then it takes off. We kind of get that brief moment of contact, but when we have that in contact for a long period of time, then it kind of tends to get a little bit muted or blunted a little bit. Otherwise, it's just kind of information overload, I think, in some ways. We kind of have to suppress some information to try and recognize the more important information that's coming through. So we do kind of have to figure out how to balance these things.
And then I just had one more question for you.
Yep.
So with the first two that we talked about, the fasciculus cuneatus and the fasciculus gracilis, those were specifically, one is sensory for the arm and one is sensory for the leg. But these anterior and lateral spinothalamic, those are for the whole body or whatever region that specific spinal nerve attaches to. So it doesn't specify like from a basic same region. Does that make sense?
So we talked about the dermatomes last time. You remember that little dermatome map with the little stripes on the person? Essentially that part of the body, wherever I'm being touched, is going to be carried on into that spinal nerve that is dedicated for that dermatome, so that we recognize pain in that dermatome area, or we recognize temperature in that dermatome's area. And so that's what would enter into the spinal cord, is based on where that spinal nerve for that dermatome comes in.
Okay.
Does that make sense? All right, so our spinocerebellar pathways, again, going from the spinal cord to the cerebellum, this is really mostly just going to be proprioceptive information. And the anterior spinocerebellar tract is more kind of the lower half of the body. The posterior spinocerebellar tract is going to have the upper. It does have some from lower, so there is some overlap between those two areas, but they're basically both carrying the same information up. That's really the main thing. You don't need to know a lot of specifics on the spinocerebellar. Just know that it's sending that proprioceptive information about what kind of forces are going through our muscles, our joints, our ligaments, our tendons, how much bend there is at those different joints. Am I in flexion? Am I in extension? Am I in rotation? That's the kind of information that's being carried on those spinocerebellar pathways. All right. So here's kind of a picture of those cuneatus and the gracilis pathways. We've got our sensory information coming in from the body, whichever region it is. The first neuron is going to be in that posterior root ganglia. Its axon is going to continue into the spinal cord and then go on up. And then if you look over to the left, this other little second section from the bottom is trying to show you the medulla. And so in the medulla, there's going to be a neuron cell body there that primary sensory axon synapses with, and then that's where it's going to cross over. It's going to decussate at the pyramids in that front portion of the medulla, and then it's going to be carried up on the opposite side through the remainder of the brain stem and midbrain. And then once it hits the thalamus, which we see in this top picture, then that thalamus is going to have an additional neuron there that says, oh, I need to send that information to the part of the brain that recognizes that this is my right arm that is getting touched. And so then that's going to synapse in that post-central gyrus to give us that sensory information and help us accurately locate and recognize in our brain, where is that on my body taking place? Where is that bug that just landed on me? So if I do want to swat it, I know where to direct that force. If I do want to be nice and pick it up and gently carry it outside, I know how to do that too. So 3 neurons, the sensory one that's getting the information from the body, the one in the medulla that's going to ultimately cross over, and then the one in the thalamus that's going to send it to its final destination so that we can make sense of where we're getting that information. Our anterolateral pathways, again, this is our pain and temperature type information, the crude touch, deep pressures. These are a little bit different. We've got our initial sensory neuron in that posterior root ganglia, just like the other one. But in this case, it's going to synapse in the spinal cord first. it crosses over at the level of the spinal cord and then goes all the way up. So before it gets to the medulla, it's already crossed over. It essentially crosses over as soon as it comes in to the spinal cord. So a little bit different, and I don't think you really need to know. There's no questions they're going to ask you which pathway crosses over where. Just know that some things, the majority of things do cross in the pyramids, but there are certain pathways like this one that cross in a different place. And this is going to be another 3-neuron chain. It's just the first 2 neurons are basically down low in that spinal cord, wherever that message is coming in. And then the final neuron is still going to be in the thalamus. So we can still send it to the correct part of the postcentral gyrus to make sense of what we're feeling. And then our spinocerebellar only has the 2. And then we don't have a diagram of it, but, oh yeah, I guess we do. I skipped over it real quick. So we've still got our sensory information coming in. Some of these things cross over at the level of the spinal cord. Some of them cross over in the medulla or the pons, but ultimately they're going to cross over for the most part and go on up. But we only have the 2 neurons, and they're both down kind of low. Again, these aren't going all the way up to the cerebral cortex where we're going to acknowledge and recognize where that sensation is coming from. It's just going straight to the cerebellum. And then the cerebellum is going to have additional connections up to the cerebrum to try and influence the motor plan that we're going to execute. So only 2 neurons in that spinocerebellar, but 3 neurons in the other sensory pathways. Now, when we look at motor pathways, those descending pathways, we only have 2 neurons. We've got the 1 primary motor cortex neuron in that precentral gyrus for whatever body part we're trying to move. And then the second one is at the level of the spinal cord, basically where that axon is going to go out to actually innervate the muscle that we're trying to activate. So we call these an upper motor neuron. If it's in the brain or the brain stem, it's a lower motor neuron is the one in the spinal cord. And so you'll often hear about people that have either an upper motor neuron injury or a lower motor neuron injury. So like a spinal cord injury, that's a lower motor neuron injury. Nothing happened to the brain, but I got in a diving accident or a car wreck that severed or damaged my spinal cord. That's a lower motor neuron injury. If I have a stroke, well, that affected the neurons up here. That's an upper motor neuron injury. Or if I have a head injury, that's an upper motor neuron injury. So we kind of see distinct problems with if the upper motor neuron's not working right, that are different than what we would see with a lower motor neuron problem. And you'll get more into those when we get into neuro class in a couple semesters down the road. Those motor neurons can either be a direct pathway or an indirect pathway. The direct pathways are going to be really the stuff coming from that precentral gyrus. The indirect pathway is going to be motor functions that are a little bit more on autopilot. It could include, it is distinct for skeletal muscle. So these are things that are going to help regulate like muscle tone and some of our more kind of autopilot functions. We talked last time about the cerebral nuclei, things like the caudate nucleus, the globus pallidus, the putamen, the amygdala. Those are those cerebral nuclei. The cerebral nuclei are really the primary things you're going to use those indirect pathways, where we can just turn on kind of an autopilot program to walk. We don't have to think about it at the higher motor cortex level. It's going to just kind of run on its own because we've done it so many times. We don't have to think about it quite as much. Or if I'm trying to stand up and prevent myself from falling over, I need to sense gravity and sense forces, but I don't want to actively make myself have to think about it. Those are the type of things that are going to be controlled with those indirect pathways. But if I intentionally want to pick up my phone, pick up a pen, punch somebody, swat a bug, kick a ball, those are all going to be direct pathway type of things. All right, so our direct pathways are our pyramidal pathways. They're going to cross over at the medulla oblongata in the pyramids to the other side of the body. When we're going all the way down the spinal cord to actually cause actions in the arms, leg, or trunk muscles, then those are going to be termed corticospinal pathways. They're going from the cortex of the brain, that perimeter gray matter, down to the spinal cord so it can synapse with the neuron there that's going to ultimately activate the muscle that we're trying to call into play. Our corticobulbar tracts refer to the cranial nerves. So these are not going to go, they're not going to go any further down than where the cranial nerve goes. And so that's kind of the differentiation. They don't really travel through the spinal column. They exit out higher up, either going to different parts of the brain or the face, or in the case of like the vagus nerve, they do go down further into the body, but they've kind of exited the spinal cord as they start working their way down to innervate the heart or the lungs or those type of things. So corticobulbar, corticospinal are both direct pathways. The corticobulbar ones are only going to be for the cranial nerves that have motor function. So if it's a sensory motor nerve that only receives sensory information, we're not going to have a corticobulbar pathway. Does that make sense? So like the olfactory nerve or the optic nerve, those just receive sensory information. They don't have any motor function. They're not going to have that pathway. But oculomotor nerve that helps us move our eyes, then that would have a corticobulbar pathway because we have to trigger some muscles to make us look left, right, up, down, that kind of stuff. All right, so corticobulbar, let's talk about our corticospinals. We've got lateral and we've got anterior. Again, these are going to be in similar regions to the anterolateral sensory pathway as well, not too far as within the spinal cord model itself where these paths are located. Our lateral ones are the ones that control limb function. And so I kind of added, and I don't know if the slides on Canvas have this in parentheses that I stuck in here. These are things that I think can be helpful to remember which path controls which function. The lateral goes to the limbs, the L's go together. The anterior goes with the axial muscles, so the A's go together. So when we're talking about controlling erector spinae, rectus abdominis, internal, external obliques, that information is going to be carried on this anterior corticospinal pathway. But if I'm trying to run, jump, kick, throw, right, those are going to be carried on those lateral corticospinal pathways because that's where the motor function for the limbs is going to be controlled. Okay, so lateral limbs, anterior for axial for the trunk. All right, how are we doing? 8:45. Okay, so this again, don't try and memorize all these little spots. Certainly, I think it's easy to remember the dorsal columns. Those are really big, but these other ones are really pretty small. Just know the general region. I don't think there's any questions that give this model and ask you to identify which of these pathways is which. You do want to know for the written exam, at least kind of which primary sensory messages are carried on which pathway. And then like that last slide, the anterior lateral, or the anterior being the axial muscles and the lateral corticospinal tracts being the limb motor pathways. The other ones that are shown on here, the rubrospinal, the reticulospinal, the vestibulospinal, and the tectospinal, those are a lot more of our kind of cranial nerve pathways, the corticobulbar pathways, a lot of things that control reflexive actions, things like that. Those are kind of the pathways that do a lot of those motions. So some of those aren't going to go really far down because they don't really impact things that are that far down in the body. So they're not going to go down necessarily to the lumbar level or the sacral level of the spinal cord because there's not really things that are controlled about vestibular function in those areas. So we'll touch on kind of the key things with those here as we go. All right, so here's our kind of our diagram of how the neurons are set up. Again, these are descending, so they're going from the top down. We're going to activate something in the precentral gyrus in our arm, our leg, our trunk muscles that we want to move. It's going to come down through the midbrain on the same side of the body, same side of the spinal cord. At the level of the medulla, typically it's going to cross over We do have some things that stay on the same side, and that's what these two different red lines are showing. One of these is going to stay on the same side until it gets down to the level of the spinal cord where that spinal nerve is, and then it crosses over. But the majority of them are going to cross over at the medulla in those pyramids and then be carried to the anterior horn to control muscle function in either the arms or the legs or the trunk, depending on if it's the anterior corticospinal or the lateral corticospinal pathway with that. And again, you don't need to keep straight which ones cross over at which level. Just know that they will cross over and decussate at some point along the chain. So, but we've only got two. We've only got two neurons. We've got the neuron up here. You can see the cell body, and then we've got this long axon going all the way down through the spinal cord, and then it synapses at the spinal cord, and that's where that second neuron is. So this neuron in that spinal cord is the one that is actually going to activate the muscle. Its axon is then going to go out and innervate that particular portion of the muscle that it's in charge of making fire. When we look at that. All right, so our indirect, again, these are going to control subconscious motor activities. So things like I mentioned earlier, postural control. Sometimes some of our reflex activities can be controlled through that. Things that we don't necessarily have to think about and be aware of and take action on, but they are still controlling spinal muscle, or excuse me, skeletal muscle. So they're not necessarily in charge of smooth muscle. So they're not visceral, they are somatic, but it's just somatic stuff that we don't want to have to think about. It's easier for our brain to just kind of turn it over to autopilot and let the body kind of self-regulate it.
So is that kind of like blinking or breathing? Like more often than not, you don't actually think about blinking or you don't actually think about breathing?
Yeah, I think so because, we do have the ability to kind of override some of these and take over control. So if we want to regulate our breathing a certain way and meditate and do deep breathing, we can control that rate of respiration. But if we don't want to think about it, then we can let these indirect pathways just kind of take over. So we kind of have that dual control ability. And that's what shows us that, yes, it is skeletal muscle. If we can control it, then it's skeletal muscle. If we can't control it, then it's going to be visceral muscle or cardiac muscle that we can't directly influence through our thoughts. All right, so here's some of our indirect pathways. Again, coming from the cerebrum, we do have some extra nuclei involved here. You can see that in this case, there's probably like close to half a dozen, maybe 4 or 5 different neurons involved. And so that's where the name indirect comes from. It's not that direct, this neuron send the message to this neuron, and then that distal neuron send the message out to the body. We've got a lot of other synapses going on here to control things and kind of help regulate things. Again, when it's indirect, if we're relying on autopilot, we kind of need to have some extra controls in there to kind of help regulate things a little bit. And so I imagine that's probably why we need these extra neurons to be involved in there, in that function. All right, so briefly, we'll talk about these indirect pathways. And I think if you just know kind of these bullet points, that's probably all you really need to know on them. The rubrospinal, means red. So there's a portion of the brain and brainstem or midbrain, excuse me, called the red nucleus. I guess it shows up being kind of red when they dissect people. And that's going to help control precise movements and some tone in our flexor muscles. So things like biceps, triceps, finger flexors, toe flexors, plantar flexors in the ankle and calf. Our hamstrings would be our knee flexors. So it's going to kind of help control those things. Again, if we're thinking about maintaining standing posture, we need kind of to balance out flexor and extensor tone to kind of keep the joints lined up the way that we want them to be lined up. The ones here in this lateral pathway, the reticulospinal, the tectospinal, and the vestibulospinal are definitely more reflexive oriented. So posture and balance reflexes in the reticulospinal, and they're called that reticulo because their initial neurons are originating in that reticular formation. That's a portion of the pons in the medulla that does a number of different things for us. One of the key things that the reticular formation does is kind of control like sleep and wake cycles and our level of alertness, being conscious and aware of things. And so that reticulospinal neurons there control balance and posture. The tectospinal, the guy in the video mentioned these. It's going to control the relate information from the superior and the inferior colliculi. Let me find my brain here. Not literally. My brain stem. So this is the brain stem. So these would be my thalamuses. Then we've got our midbrain, we've got our pons, we've got our medulla. This is looking at the front. If I turn around and you look on the posterior side, we've got these 4 little bumps kind of right here in the midbrain. And that's close to the if you remember the epithalamus, it's basically where the pineal gland is. It's going to be just below the epithalamus is where these clusters of neurons are. These are going to be the superior colliculi and the inferior colliculi. These are involved in hearing and vision reflexes. So if you're outside or whatever, and all of a sudden you see something flash by in the corner of your eye, our eyes will kind of reflexively shift to track that object. That's a reflexive response, and these are where the neurons are that control that. If you're like me, I go on a lot of runs and hikes and stuff in the mountains, and I'm usually by myself, or maybe I have the dog with me. If I hear a twig snap, then I whip my head around and I'm like, is that mountain lion about to pounce on me? Or is that moose about to trample me? That's the hearing reflex. Essentially, that's a protective mechanism. If we hear something, we're like, what was that? We whip our head around really quickly to sight it. That's kind of where these neurons are going to be located that control that. So these are protective mechanism reflexes to kind of keep our whole person alive. So we recognize if something is coming at us, somebody's swinging a bat at us, we want to pick that up early and reflexively know, there's that bat, I better move and react to it. We need that kind of stuff.
For those that are paranoid, are those reflexes enhanced?
Could be a little
Okay.
hypersensitive. You might be a little bit more tuned into them. Some people kind of like, the outdoorsman guys like, I'm not going to worry about it. I hear that crack in the woods all the time. It's never anything big. And maybe they let their guard down, and other people are more hyper aware of it potentially. So yeah, you have different, and that's probably related to the reticulo activating system too. Kind of how tuned into your environment are you? How alert, how aware are you? They're all going to be kind of interrelated that way. And then vestibulospinal tract reflexes that help maintain balance. We'll talk about our vestibular in lab in particular today when we go over the ear, since we've got that function there. All right, 9 o'clock. I'm not doing too bad. So let's keep rolling here. All right, let's look at our, where was I? Cerebral nuclei. That's what I wanted to talk about. So the old term was, when I was in school, we called these the basal ganglia. And then somewhere along the line, somebody said, well, ganglia are really more peripheral clusters of cell bodies. So they're really nuclei, not ganglia, if they're in the CNS. And then they changed the name from basal to cerebral. I'm not really sure why. But these are in the deeper portions of the brain. Again, these are going to be those things like the caudate nucleus, the putamen, the globus pallidus, the amygdala. Those are our primary kind of cerebral nuclei. And again, some of them control kind of repetitive functions like walking that we don't really want to always have to think about. Once we've mastered the task of walking on a level surface, we can just kind of fire up these neurons and it'll help us get from point A to point B. We still need to be, our body still needs to receive central information about uneven surfaces and make adaptations. If we need to adjust our step length to step up on a curb. So we don't just give up complete control of it, but it lets us not have to think quite so much and expend so much mental energy to think about walking from point A to point B if we let these cerebral nuclei take it over. I think it's the caudate nucleus that controls that particular thing. So this is kind of an interesting view of the brain. They basically have cut away the left posterior cerebrum, the left posterior or the left cerebellum as well, and then kind of left half the spinal cord or brainstem visible in there. So again, these are going to be fairly close to the thalamus, all of these cerebral nuclei, kind of either right next to the thalamus, right below the thalamus, but very close to that central portion of the brain, close to that diencephalon region that we've got. Their messages do need to get sent down through the spinal cord, and we have that reticular formation, again, that is in the pons and the medulla, the vestibular nuclei, that's cranial nerve 8. So that's going to be where the neurons for cranial nerve 8 are kind of located, so that we can make sense of changes in our head position and body position and speeds that we're traveling at and things like that. So that's kind of where they're located. You can see the tectal neurons back here, the superior and inferior colliculus, and then the red nucleus is kind of just in the midbrain. It's kind of what this area is looking at. So that's kind of where these things are located. All right, so cerebellum. So cerebellum is our balance, kind of fine-tuning center in the brain. So it definitely plays a huge role in coordination, balance. It's going to try to help us receive information from the body so that when we then try to generate a motor plan through the precentral gyrus neurons, we can kind of tweak it and modify it to improve its accuracy and make that movement be better, more functional, more help us keep our balance or make our motions more skilled and go where we want them to go. So we have this, what's called an error correcting signal. Essentially, the cerebellum senses what position we're in. When the motor neuron tries to send its general message down, the cerebellum's like, no, wait a minute, we need to modify that plan a little bit, because if we just do the basic throwing motion, let's say, that's not going to work, because you fielded that ball while you're leaning way over and you're not throwing from the same position. So the sequence of which muscles need to fire when is slightly different. So we need to correct that gross motor plan to fine tune it a little bit. And so that's really kind of how that cerebellum works. It's got some fairly complicated pathways. I kind of like this slide because it gives you a key over here to kind of let you know which arrows represent what function or what message. So initially we've got this blue one coming into the cerebellum, receiving information from the body that's trying to tell us from the proprioceptors in those, that Golgi tendon organ, the muscle spindle, how much tension is going through that muscle, how much stretch do we put on it, so that we know where we are in space, how bent my knee is, how much pressure is on the ball of my foot, that type of stuff goes in there. The pons also is going to feed in some information to it. And then the cerebellum needs to kind of figure out what it needs to do about that. So these green ones are integration and analysis. It's comparing what is that gross motor plan, how is that going to match up with where my body actually is, and how do I need to tell those motor neurons to fire to kind of change their sequence of firing a little bit. So that's kind of what the yellow arrow is. The yellow is kind of that correction thing. The red ones are saying, this is what I want to do. The cerebellum says, well, hold on a minute. Let's modify that a little bit. Here's a better way to tweak that motion. And so it needs to get that information from the cerebellum to really be accurate with that. Did I tell you guys, I told you guys about my patient when I was a student that got shot through the cerebellum, I think. Seeing some nods, seeing some not nods. He was in the back seat of a car and got shot in drive-by shooting. And it took out one half of his cerebellum. The other half was totally fine. He had perfect coordination on one side of his body, but could not reach out to touch a finger or anything with the other hand. If I held my hand in front of him and had him try and reach for it with that hand, he could make the muscles work because the cerebrum was not affected, but he could not guide that motion and accurately place that finger. And when he tried to walk, he could not accurately place that foot down where he wanted it to. So he had a huge risk for falls because he just couldn't integrate the sensory message that he was getting in and use that information to then direct the motor action to cause coordinated movement. It was a really insightful patient to see just because it was a pretty unique injury that he had. All right. So simple reflexes. First is brain complex reflexes. Our simple reflexes are going to be kind of like we had you guys try and test the last time. Things like that plantar reflex, or excuse me, the patellar tendon reflex, things like that are kind of your basic simple reflexes. It's not going to go up to the brain where the brain really has to make a lot of sense of it. Things like when we talked about those tectal plate neurons, the superior and inferior colliculi, those are a little bit more complex reflex. We need to be able to not only kind of hear something or see something, we need to be able to react to it quickly and then also make sense of what we're seeing so that we know if we need to do something else to take evasive action from that thing swinging in at us or that mountain lion about to pounce on us or whatever the case might be. So it's a little bit more to it, a little bit more complicated pathway when we get into these more kind of tricky reflexes that call into play more different parts of the brain. All right. Let me skip that one. I don't think that's super critical. I wasn't going to go over that slide. I don't want to confuse you too much. I already am, I'm sure. This is not easy stuff. The brain's not easy. So don't feel bad if you're kind of lost a little bit. You're going to hear it again in neuro as well as other classes too. So hopefully by the time you're done, you'll have a better handle on things. So higher order mental functions, things that we have to think about, something that's going to involve that frontal lobe where we have really most of our cognitive, executive planning, kind of making sense of things is up in that frontal lobe. Certainly the other lobes are important and we're going to have communication between those lobes to kind of help do things well. But that's kind of the main area that we need for that higher order thought processes. We've talked about hemispheric lateralization, how certain parts of the brain control certain parts of the body, how certain one half of the brain controls the other half of the body, those type of things. But they've also identified that really the left side of the brain is much better at doing certain things and the right side of the brain is much better at doing certain things. When people have a brain injury that affects one side of the cerebrum or the other, we have a limited ability to adapt depending on which area gets impacted. And we are not totally symmetrical in the brain, depending on if you're right-handed or left-handed. That's kind of what this slide is trying to show you. For the right-handed person, their right frontal lobe is a little bit more prominent, but their left occipital lobe is a little bit more prominent. So it's a little bit, like I said, we're not totally symmetrical. And it makes sense because we do have a hand dominance that would require more neurons, either to have better sensation in that hand or to have better motor function, and especially fine motor control in that dominant hand. All right, so cerebral lateralization.
What about a person who's ambidextrous? For example, I can write equally well with both hands. So what is happening there?
You're special, I guess. I don't know for sure how that evolves in people. Certainly habit can be part of it. My wife and I are both left-handed. Our son is right-handed. But like he still brushes his teeth with his left hand because when we were teaching him how to brush his teeth, it was far easier for us to stand behind him and help guide the toothbrush with our left hand. And so that's kind of just how he developed that skill. So he still, well, he has an automated toothbrush, so it doesn't really matter anymore that much. He can cheat. But he writes with his right hand. He's far better at throwing a Frisbee with his left hand than his right hand, but he'll throw a football or a baseball with his right hand. So he's kind of ambidextrous, but part of that I think is more of a nurture thing because his parents were both left-handed, that led him to do certain things like we did. I don't know if that's the case for you, Tom, if you had one parent that was right-handed and one that was left-handed, or if at an early age, maybe you did certain tasks that required fairly equal use of both hands that might have given you that ability, or it might just be the way that you're wired for whatever reason, but I'm not sure why some people have more ambidextrous skills than others. Certainly if somebody loses a limb, or if they have a brain injury at an early age, we have a better ability to adapt when we're kids and kind of change those plans and get better at utilizing the non-dominant side than if we're 20, 30, 40 and we have an accident like that. It's a little bit harder to make that transition to get a dominant hand, to lose a dominant hand function and let the other side take over that role. It's a little bit harder when we're older.
So there's the rest of the story for me then, because I'm from Ohio, rural Ohio. And as a kid, from the area I came from, people who were left-handed were thought to be slower than people that were right-handed. And so the story I got as a kid was that my grandmother would correct me anytime she caught me using my left hand.
Yeah.
And so I grew up using my right hand, and when I was 14, I broke my wrist really bad, and I had to have a cast that encompassed my thumb for almost 5 months. I couldn't use my right hand to write, and I started using my left, and it didn't take long at all before I could write fine with my left hand. So I don't know if I'm right or left-handed, but that's the story.
Yeah, I mean, it could very well be that you were genetically programmed to be left-handed, and then you were conditioned to be right-handed, and that was a fairly common thing. There's actually terms for, kind of alternative terms for being left-handed and right-handed, and to be right-handed is called dexter. To be left-handed is called sinister. So it's not really a very positive term. And if you knew anything about the TV show Dexter, he was a pretty sinister character. He was like a serial killer. I had a friend of mine that was actually a cameraman on that show. But yeah, it's certainly being left-handed was not acceptable at certain levels, and a lot of cultures too. The left hand is what you use to wipe your butt with in a lot of cultures, and so to use that left hand for anything else was like a horrible thing to do. So there's a lot of reasons why that might be, but in your case, it could be that you're conditioned to be right-handed, and then when you're faced with a challenge of using that right hand, it was easy for you to do that. My wife is left-handed, and she broke her left wrist and had to write right-handed. She was taking some classes at the time, kind of like you were, and the sad thing is her handwriting was still way better than mine, even when I was using my left hand. I could write faster. She was slow. It was much more deliberate. She had to think about it a lot more because it wasn't a skill that she had developed, but it was still way more legible than my writing. So pretty sad. All right, let's talk about this picture a little bit and kind of look at some of these things. We'll talk about the vision a little bit more in physiology. Our eyes, and I know Mo asked me about this, I think last week. We have our optic nerve coming back from these eyeballs, and you can see the green and the purple coloration. Essentially, we have what we call our medial field of vision, what we see kind of closer in towards our nose, and then our lateral field of vision, what we see more out to the sides. And that information is actually carried to the back of our brain in a different way. But when light is coming in from the inside, kind of at this kind of angle and moving this way, it's going to be picked up by the lateral retina neurons. That information ends up basically being carried back on the same side of the brain as that eye. The stuff that we're picking up, if light's coming in from the lateral side and projecting in medially, the image that we pick up on the inside of that retina is going to actually cross over and will be interpreted on the opposite occipital lobe. So both the right eye, half of that field of view goes to the right side of the brain, the other half goes to the left side of the brain, and then same thing with the left. When all that information is carried together from one eye, that's called the optic nerve. When we get to where it crosses over, we call that the optic chiasm. then that nerve, as it continues on and carries that information to the occipital lobe, we call it the optic tract. So it changes from the nerve to the tract because the tract carries messages from both eyes, whereas the nerve carries just the message from the one eye that it picked up initially. The other big things that we look at with the hemispheres, the left side of the brain is really more our kind of objective side of the brain. The right side is a little bit more of our artistic side of the brain. So we call this categorical hemisphere as the left, and the representational hemisphere is on the right. So if we look at in these frontal lobes, we've got verbal memory. So when we're trying to read and write and speak and understand those things, that's pretty much all on the left side of the brain for most of us. The right side, memory for shapes. So if we're trying to feel, recognize, identify triangles, circles, ovals, rectangles, that type of information is much better on the right. Things like singing, musical instruments, creating like poetry and stuff like that, that's a little bit more on that creative side, that those are generally attributed to be the right side dominant person. People that are really good at math, things like that tend to be, and just critical reading and things like that tend to be a little bit more on that left side of the brain. We have both the Wernicke's area, this yellow area, and the Broca's area, that blue area, that are both on the left side, and those are really more of our visual, our verbal communication, our written communication. All that's on that left side of the brain. But again, the right, musical and shapes and feeling and things like that. I was trying to pick out videos to show you guys today, and I watched another one that was talking about a patient that had a stroke that affected the left Broca's area. And so that person lost the ability to speak because they could not coordinate the neurons there that would generate mouth and tongue movements to create words that made sense. And they found over time that if the person would just sing what they wanted to say, they could get the words out, because the right side of the brain had not been affected, and essentially where the verbal communication for that artistic medium of singing was still working. That was the way that that person ultimately became able to communicate verbally with his other people around him. And over time, they learned how to modify that area of the brain to actually speak, where they didn't have to like sing what they wanted to say. They could actually get back to speaking. But the right side did take over that ability, because it does have, you know, common functions, but they are just tweaked a little bit differently. So it's kind of an interesting case of kind of how the brain can make some of those changes sometimes, depending on exactly what's been damaged or affected up there. All right. We do tend to see some differences sometimes between the genders, and it's not like a hard and fast rule, but a lot of times if you think about like traditionally men are reluctant to ask for directions. You're driving and you're like, oh, I can figure it out. Yeah, we just need to take a left up here and do that. And that's kind of the stereotype type for the man. And then the woman that's the passenger is always like, yeah, I told you you should ask for directions. You were lost. You didn't know what you're talking about. You didn't want to admit it. But that spatial awareness is one attribute that they tend to kind of associate with men more. Whereas with women, they talk about women's intuition, that they just kind of have this sense of what's right to do, or what would be a good idea or a bad idea to do in this scenario. So we do have these certain attributes that people believe are more closely linked to one gender or the other. Again, there's always exceptions to the rule, but I would definitely say I have better spatial awareness than my wife. We'll be out for a hike and she'll be like, oh, I don't know, where do we go? And I'll be like, well, the mountains are this way and this is this way. And I'll be like, and she's like, oh, okay, yeah, that makes sense. But it just doesn't click for her. My wife's got a PhD. She's very bright. But we both have different things that we're better at than each other. But I better be quiet before I'm outnumbered by the ladies here. So I better just stop off my head. All right, so we've already talked about Wernicke and Broca's areas a little bit. Again, Wernicke's is kind of back here, kind of close to where the temporal lobe, the parietal lobe, and the occipital lobe kind of all meet. So the things that we're hearing are coming in through that temporal lobe. The things that we're seeing on a piece of paper and reading are coming through the occipital lobe. And then in that Wernicke's area is where we kind of make sense of what we're hearing or reading. And then Wernicke's area has to communicate with Broca's area, which is more anterior in that frontal lobe, so that we can actually control mouth, tongue movements to form the words that we're after. And when people have damage in those areas, either Wernicke's or Broca's will have distinct problems that show up in their speech. So whether they had a brain tumor or they had a stroke that deprived blood flow to these neurons, those type of things, or a head injury, those things would damage these particular areas. And we'll see either apraxia or aphasia. So apraxia is a motor problem. That would be more common with like Broca's area problem. If I cannot give the motor commands to form my lips and tongue in a certain way, I'm not gonna be able to get the words out. So apraxia is much more of a Broca's problem. Aphasia is much more of a Wernicke's area problem. Aphasia means you cannot understand what you're saying, or maybe you understand it, but you can't send them the message correctly to Broca's to form those words correctly. And then within aphasia, you can have either what's called receptive aphasia or expressive aphasia, or you can have global aphasia. So receptive aphasia is the difficulty understanding. I'm hearing something said, but it doesn't make sense to my brain, so I don't really know how to respond to it. If you have expressive aphasia, you understand what's being said, but you can't get the words out to respond appropriately. If you have global aphasia, it's kind of both. I can't make sense of what's being told to me, and I can't form a coherent sentence to say anything meaningful either. And so these people might be able to form words, because Broca still might be working, but those words are not in the correct order or the correct context. They're just kind of more randomly generated. So kind of different. If it's apraxia, then it's really just that you can't, that's what can be like slurred speech, things like that are going to be more apraxia. Who had a question? I heard it.
Can they write them? So if they write it down, it'll be right?
A lot of times, yeah. So you can use things like writing. So if they have receptive aphasia, or excuse me, if they have expressive aphasia, they can understand what's being told to them, and they could very well be able to write down their responses or point at a word board and communicate that way. So it's definitely a better situation to have expressive aphasia than receptive. If you have receptive, it's really, really hard to communicate with that person, because anything you tell them, no matter how simple you make it, it still might not make sense to them. And they still might not be able to follow commands of what you're asking them to do. So here's those areas. Again, kind of right close to where parietal lobe, temporal lobe meet, and kind of close to occipital lobe. That's going to be our Wernicke's area. And then close, kind of this anterolateral portion of the frontal lobe is that Broca's area right in there. All right, let's talk about memory just a little bit. Memory is going to involve a lot of different lobes depending on what type of memory we have. And many of our memories are multi-lobal, where we have a visual memory of what we saw, we have an auditory memory of what we heard, we have a olfactory memory of how it smelled potentially. All of those things are going to kind of come into play. So we can definitely have sensory memory. This is generally pretty brief. And this is really important for us when we are developing motor skills. If you think about learning how to throw or kick accurately, whether you're throwing a ball or a dart or whatever, let's say I'm throwing a dart at a dartboard, and I throw that dart and I'm trying for a bullseye, and I'm like high and to the left. I see that it's high and to the left. I recognize sensory-wise where my hand was when I let it go, how my arm was moving across my body as I released it, and I try and make some corrections to that motion to improve the accuracy of it. But if I don't do it right away, that sensory image and that sensory sensation of what I saw, what I felt internally is lost. So when we're trying to improve those motor skills, that sensory memory is really critical, especially the proprioceptive part. But we definitely need the visual part too, so that we can understand what was wrong with our motion. Why was it going to the left? Why was it going high? Why was it going right? As far as not hitting the target that we were after. So that's the first kind of type. And that's going to definitely be related to kind of what we think of as our muscle memory. If we keep working on that skill and eventually figure out how to execute it with an accurate fashion, if we keep doing it accurately, then our brain starts creating this motor memory that becomes more kind of like those cerebral nuclei. We've done it enough times that it becomes more autopilot. Think of like maybe shooting a free throw, something that's a highly repetitious act. If we get ourselves in the right position and minimize the variables and try and be really consistent in how we set up for that shot, then that motor plan becomes simpler to execute because there's not as many moving parts. And that can be really helpful. Short-term memory is what a lot of people do when you cram for an exam. That information sticks for a little while, seconds to hours. But if I ask you about that concept next week or 2 weeks from now, it's gone. It never got converted over into long-term memory. So we have to work to get short-term memory converted over to long-term memory to make it stick. So I was dissing on my wife's directional sense, but now I got to go the other way and tell you where she's way better than me. We took some classes together. We did our master's in kinesiology at the same school at the same time. I was a year ahead of her, even though she's a little bit older than me. And even now, whatever, we met in the late 80s. So it's been whatever, 30 something years since we took those classes together. She'll remember tons of detail about these concepts. And I'll be like, yeah, I can remember the big picture, the general stuff. So her long-term memory is way better than mine for a lot of things. My short-term memory, like my light bulb goes off a little bit faster than hers. I can be presented with fewer pieces of the puzzle and kind of figure out the answer, whereas she needs a little bit more of like all the information. But once she has all the information and has a chance to think about it, it's going to stick, and it's going to stick forever. Whereas me, I was good at taking those tests, but 2 years, 3 years down the road, or like anytime I teach this class, I have to review everything before I teach it. Certain things I remember, but certain things that I don't use on a regular basis, I have to constantly reinforce. So my short-term memory and my ability to pick stuff up quickly is, I would say, better than hers. But once it gets into her brain, it stays. And in my case, it kind of tends to disappear a little bit too. All right. So consciousness. It's more than just being awake and or asleep, but that's kind of the big kind of differentiation. And again, the reticular formation is the part of the brain that helps control that. So if you wake up to an alarm, the reticular formation in the brain is kind of the first part of the brain that would be recognizing, hey, that thing's beeping in the background. Aren't you supposed to be awake now? And then it sends a message to the cerebral cortex. It's like, hey, don't you hear that alarm? Wake up. And the brainstem, those pons and medulla is where that reticular formation is. That's what kind of helps us come to and recognize, oh, okay, I do need to get up. That alarm is going off. I better shut it off. So reticular formation, sometimes called the reticular activating system. That's a particular part of it. But it is not only responsible for conscious awareness, but also kind of kicking in those reflexes that control muscle tone and posture. When we're lying down on a bed and sleeping, we don't really need muscle tone. We're not fighting gravity. The bed is supporting our whole body. But when we're upright and awake, we need that resting muscle tone to keep us from just slumping over in the chair or from getting that head nod. You know, you're trying to stay awake in class and that's kind of, you're losing that awareness and that head is dropping. And then we get that postural muscle tone that's like, hey, your head is tipping forward. Why don't we activate those extensors and we correct it? So that's when you're kind of half awake and half asleep. That reticular activating system is not quite keeping you totally alert. in that case. So these are going to be tied into that sleep-wake cycle. We think about circadian rhythms, things like that. Those are kind of related to hormonal releases and different things like that. All of that's kind of really important and related to this reticular formation portion of the brain. So again, in that pons in the medulla, in the brain stem, that's kind of where we get that information. And it does need to ultimately get up to the cerebrum where we are conscious and aware in the thinking part of our brain. So that's its job to kind of keep us engaged, keep us awake, keep us involved. All right. Many of you may have heard of an EEG. We'll talk about an ECG coming up in a couple of weeks when we go over the heart, but an EEG is looking at brainwave activity. You're essentially looking at electrical activity caused by neurons depolarizing and firing, and we can learn a lot about how somebody's brain functions and look for problems when they use an EEG. I had a suspected seizure a couple summers ago and had the pleasure of doing a 72-hour EEG. So I had to have these electrodes on my head for 72 hours. And it wasn't fun. Fortunately, it was between classes. So they didn't make me come into work. They let me work from home because I wasn't able to shower or do anything like that for 3 days. It was not fun. They didn't end up finding anything. They never really could figure out what was going on. But I was not allowed to drive for basically a month until I got all my tests back. I had to go get an MRI done. I had to do a CT scan, had to do this 3-day EEG to clear everything before the neurologist would say, okay, you're safe to drive. You're not going to have a seizure while you're driving. We don't really know what caused that episode, but when people have seizures, obviously you don't want that person driving if they're going to seize and not be able to maintain control of their vehicle. And so EEG is one of the things that they use to identify that. They'll use it a lot for identifying sleep disorders as well. So there's a lot of other reasons why we might do an EEG. And then certainly when we look at people that are in a bad accident and they're like, oh yeah, they're brain dead, that's what they're basing it off of. The EEG activity shows that they're not really having conscious thought anymore. Their brain stem is still keeping their heart going. It's still keeping the respiration going, but everything upstairs isn't working right. And so that's when, you know, you could keep that person alive on machines potentially, but they're never going to come out of it. And so the EEG is the tool that they're going to use to kind of diagnose some of that stuff. All right. I don't think there's any questions on sleep, so I'm going to skip over sleep here. I think that's it. Yep. So 933, any questions on what we covered in that chapter? It's kind of a mix. The first part of that chapter goes over those pathways in general, and then the other part kind of tries to tie in different parts of the brain that we've already talked about a little bit, and how those pathways kind of influence and how they all kind of interconnect. And again, it's those axon connections between different parts of the brain, between the sensory system and the motor system, and the parts of the brain that kind of help balance those things reflexively. It is tough to keep all straight in your head. I'll look over the exam on this content, and when we get a little bit closer to it, we'll try and help review a little bit what's the more critical stuff. I think the study guide that's up for that next test probably gives you a little bit of guidance already about which things to focus on most heavily. And our quiz next week won't be over this. Our quiz next week is, like we talked about last time in lab, is just over the eye structures and the ear structures. So if we don't have other questions, let's take about 10 minutes, and then we'll come back and we'll get into the ear, kind of go through those, and then we'll just see how much time we have left for lab once we get through going over the ear. So take about 10 minutes, maybe 9:45, we'll start back up. So Mo, you got all those eye structures down? Committed them all to memory already?
I hope so.
There you go. You got some more days to get it down.
Where is that in our book?
The eye and the ear, I want to say it's chapter 19. So we're on chapter 7. We just did chapter 17. So we'll do autonomic nervous system on Tuesday. And then I think next Thursday, we'll probably go over the senses chapter, which has the eye and ear in it, but then also talk about sense of smell and sense of touch, sense of taste as well, and kind of how those are all set up.
So why do we jump ahead?
I think lab wise, it's a little harder to, I didn't set this course up initially, but the way that the labs were set up were to try to focus more specifically on the areas that we in PT would treat and address. And so it's not that we wouldn't treat or address brain structures, it's just a little bit harder to, I don't know, it's all so conceptual. Whereas the eye and the ear are more structural. We've got these key components, and we can identify certain parts of vision dysfunction or hearing dysfunction that we might need to treat or help somebody figure out how to move themselves better when they are deficient in some aspect of their vision or some aspect of their hearing. It's to make it harder for you guys. No, it's not. That's not the purpose. I think it's just kind of the way it is. Same like when we get to the endocrine chapter, which I think is chapter 20. It'll be the first chapter on the last small test. We don't really have a lab activity for the endocrine system. We kind of jump into looking at the heart more specifically and the lungs more specifically and spend a little bit more time on those two organs. So we just, like I say, we just try and make the labs be based more on things that we're more likely to kind of treat and have an impact on in the field of PT. All right, looks like it's about that time. So any questions on the eye from anything that you might have looked over since we talked about it on Tuesday? What certain structures are about or kind of how it works? We'll look at a little bit more on eye function, like I say, when we go over the senses chapter. And I think we'll talk a little bit about like those visual fields I was mentioning when we talked about kind of how the brain is set up and the optic tract. So we'll go back into this information when we get to chapter 19 next week. But the quiz next week on Tuesday is really just on all those different eye components and then all these different ear components. So let's talk about the ear. I think I'm still recording. Yeah, still recording. Okay. So we've got our external ear, we've got our middle ear, and then we've got our internal ear. And I've got a little model here. That's a pretty good model. It's, I think, fairly close to life size. I think the ear is maybe just a little bit small, or maybe I've got big ears. I think this is, but it's pretty close to life size. So the external ear is essentially what we call the auricle or the pinna. Has two different names. That's basically just the part, the fleshy part that we can touch and feel. And then it also is going to include what's called either the, you'll see a couple different terms. I think this book calls it the external acoustic canal. Some will call it external auditory canal. Some will call it the external auditory meatus. A meatus is essentially a canal. So you'll hear some different terms. But the external ear includes basically everything up to the eardrum. Once we get to the eardrum and we move to the other side of the eardrum, what's known as the tympanic membrane, then we get into the middle ear. So on this particular model, let me make sure I don't, right there, that little whitish structure, that's meant to be the eardrum. Let me make my slide smaller so you can see this. Stop sharing the screen. So this will be a little bit bigger for you guys. So we've got our auricle or a pinna. We got that external acoustic or auditory canal. And then when we hit that eardrum, then that's kind of the start of the middle ear. The middle ear is kind of the smallest part. It has the eardrum, and then it has three little bones that basically help transmit sound from the eardrum to the internal ear, the inner ear. And I think I'll just go to the slide so we can kind of look at those structures a little bit, and then I'll show you some more things on the model when it's appropriate. So we'll pop back over to that. All right. So all three of those parts have to work together. So this slide is basically kind of what I was showing you before. We've got our auricle or our pinna. We've got our external auditory canal, and then we hit the tympanic membrane or eardrum. Beyond that tympanic membrane, then we have the middle ear. So you can see up at the top, the blue is the external, this more pinkish color is the middle ear. And so that middle ear ends with a couple different membranes, essentially. We have a little membrane here that's called the oval window, and then we have another one down below that's called the round window. Other than those two windows, the middle ear is essentially the eardrum, and then these three little small bones. These are, I believe, the smallest bones in the body. We have the malleus, also referred to as the hammer. We have the incus, also referred to as the anvil. And then we have the stapes, also referred to as the stirrup. So those three bones basically help transmit the force. Sound waves coming into the ear are going to hit that eardrum. The eardrum is going to oscillate and move depending on how loud, how forceful that is. And then that motion gets transmitted from these bones that essentially have joints between them. So that if I move the malleus, then the incus has to move. If I move the incus, then the stapes has to move. Where the stapes connects to the oval window, this is an air-filled space, the middle ear. When we get to the inner ear, then we have more of the gelatinousy liquid inside the internal spaces. And so that movement of air that's being transmitted from the eardrum to those three bones then gets transmitted from that oval window as the stapes is moving back and forth and pushing that membrane. When that membrane moves, then that's going to move the fluid that's in that inner ear. And when we're talking about hearing and sound, those waves are going to be transmitted into the cochlea. So the cochlea is this snail-shaped, kind of curly, looks like a shell section of it. And so that's where we interpret sound. This other arrangement up on top, these three little rings, and then we've got a couple other little areas too. Those are the part of the ear, the inner ear, that help us with our balance and our equilibrium. So the vestibular portion. So when we learned about the cranial nerves, we had the vestibulocochlear nerve is cranial nerve 8. These branches that are coming off, these yellow branches that are tying in on this more looped structure, are the vestibular portions of that nerve. The bigger branch that's coming down and tying into this little snail shell, the cochlea, that's the cochlear branch of that nerve. So it enters into that space as one big nerve, but then it deviates so that we have the specific branches that are in charge of the cochlear function and the part that's controlling impulses coming into the vestibular portion. And so we'll look at kind of all those and how they work a little bit. Other things that we have in that middle ear, if you continue inferiorly, we've got this tube coming through, and this book references it as the auditory tube. Sometimes you'll hear it called the eustachian tubes. If you've ever heard about like little kids having to get tubes in their ears because they get chronic ear infections, that's the area that they're trying to drain, essentially. If you've flown and your ears pop, that's essentially a pressure differential. This auditory tube, you can see at the bottom, it goes to the nasopharynx. So it goes to our sinuses. And so that's why when sometimes if you blow your nose really hard, you'll feel your ears pop. When you swallow, and you're flying, you can hear your ears pop, or you're driving up on a steep mountain road and you get into higher altitude and you need to kind of change that pressure. That swallowing motion kind of helps even out the pressure between this middle ear space and those sinuses. But it is one continuous connection between those areas.
So when you go swimming and you get that water sound in your ear, is that in the external acoustic meatus?
I think, yeah, for the most part, it's going to be in there. That'll drain out, but I think you can maybe get a little bit of leakage maybe into that space, and that might cause it. I know if you put, like alcohol drops in your ear, you can kind of dry it out a little bit. It'll help it evaporate a little bit faster and clear it. But yeah, you probably are getting some sort of communication in there. Certainly when you dive deep, like you go down to the bottom of the pool, sometimes your ears will hurt if you're 10 or 20 feet underwater. And that is related to that pressure differential in those spaces, causing some of those changes.
But the water, when you're swimming, that water in your environment is not getting past the eardrum, correct?
It shouldn't, unless you have like a perforation in that eardrum. It should be pretty much a watertight seal, but obviously you do get that sensation of having some ear in there, even when you turn your head, kind of shake it out. And so I think in those cases, you might get a little bit that gets into that auditory tube and might still be lingering in there a little bit. All right, let's look at some of these structures a little bit more up close and kind of see how they're put together. So the external ear is pretty basic. It's just the part that kind of collects sound and funnels it into that external auditory meatus. And then once we hit that tympanic membrane, that's your eardrum. You do see some muscles in here. And so this tensor tympani muscle and this stapedius muscle are really important in helping protect the inner ear structures. If you've ever been to like a loud concert, you come out of that concert and like you're having to shout to the person that you're talking to. There's no music going on anymore. The band left, but your ears have kind of changed their calibration. And essentially what happens is if we have really large, big sound waves coming into the ear that are causing that eardrum to move a lot, we don't want it to just tear and rupture. So those muscles will essentially reflexively create some more tone so that it makes it harder to move the bones that are attached to that eardrum. And if it makes it harder to move those bones, then it makes it harder to transmit that force over at the stapes to get into the cochlea. So it's protecting the cochlea. And after you've been outside of that loud concert for a few minutes, these muscles recognize, oh, it's not loud anymore. We can go back to our normal setting. We don't need to like hold everything tight so those things don't move too far. So that's kind of how that situation occurs. It's basically just an increase in muscle tone of these two sets of muscles that are basically keeping those bones from moving too much. If we didn't activate these, then that loud noise, like if you have a really loud noise like that catches you by surprise, maybe you're shooting a high-powered rifle and you don't have ear protection in, and all of a sudden a big sound, then you can not block it. Your muscles might not react quickly enough to respond. But when it's more of a continuous high noise, like an hour and a half in a loud concert hall, then the ear's like, okay, we got to do something about this. I'm going to get overwhelmed and I need to kind of modulate that force of air movement a little bit. So that's kind of how those work. Now, I don't really see the hammer that much. I don't really see the anvil that much, but that's what the nicknames of these bones are. But the malleus is this first one that's attached directly to the tympanic membrane. The incus is the middle one. And then the stapes, it does look kind of like a stirrup. So that one makes a little bit more sense visually to me. And the way I kind of envision the stapes and how it works with the oval window is if you think about, like, say you had a balloon or some section of latex that's very flexible membrane, and you have like a suction cup dart on it. And if that suction cup is held on that membrane, I can pull it back and that membrane is going to give, or I can push it in and that membrane is going to push in. So essentially that stapes is the key one that's going to transmit that force to the fluid in the cochlea to let the receptors in the cochlea actually interpret what that sound means. So the round window is kind of where that enters in, or excuse me, the oval window. The round window is essentially where the sound waves are going to leave the cochlea. So they kind of dissipate. So that's basically like our release valve. And it's not it doesn't have like a flap that lets the sound waves out, but it is a membrane too. So they're both membranes. So if I push in on the oval window, then the round window ultimately is going to kind of push out to kind of release some of that pressure. Otherwise, as more sounds come in, it would just kind of keep rattling around in the cochlea and would make it really hard to interpret what is the current sound that I'm hearing. So the round window is key for kind of dissipating sound waves so that we can be able to receive more sound waves coming in to the oval window so that we can kind of make sense of what we're hearing. So both of those structures are pretty important. All right. The inner ear is the trickiest part. It has the most to it, and is kind of the hardest to understand. Is there a question? I heard some background. Guess not. All right, so we've got these things that are called labyrinths. And a labyrinth is kind of like a maze, essentially, just a convoluted space with a lot of twists and turns that's kind of hard to navigate. And so we've got what's called the bony labyrinth and the membranous labyrinth. The bony labyrinth is essentially the structural bony part of the skull where all these structures are housed. The membranous labyrinth is where we actually have the receptors that help us interpret either motion if we're talking about the vestibular portion, or sound waves if we're talking about the cochlear portion. So those two labyrinths are pretty important. So here's kind of a blown up look of those inner ear structures. And I'm going to take it out of this model real quick. This is a pretty cool model. And so I do wish you guys, we could have had lab on site so you could see this and handle this. If I can, I was trying to hold it up and take it off. I think I'll just take it off and then show you. So again, this model is fairly close to life size. I'm going to pull what you're looking at on the computer screen out. I'm holding it by the nerve itself. So I'm holding it by the vestibulocochlear nerve. This is the whole thing. So you can see my fingertips and you can see how big that is. There's the little loops. And then there's the cochlea part on the bottom and the inner ear part on the vestibular portion on the top. So it's tiny. This is, pretty darn small when we look at it. Within the skull, where we're talking about is essentially this area here is what we call the petrous part of the bone. So we've got the bigger part. This is the posterior part. This would be where your cerebellums would sit. So just in front of the cerebellum, then we have foramen magnum. So essentially on either side of foramen magnum is where all these structures are housed. If I tilt this, you can see where the hole is for the ear is right there. And so then if I hold it like this way, you can kind of see where my finger is coming in and where that bony section is. So that's where all this stuff is housed within the skull. But it is pretty darn small when we look at those structures. So let's look at the cochlea first, because I think that's the simpler of the two of these pairs of structures to kind of look at. So we've got this kind of bony shell that everything is housed within. And then within that shell, we've got these distinct membranous areas. And inside all of these areas where you see this pink coloration, you're going to have a gelatinous type of fluid in there that moves. And that's what sets off the receptors, is the movement of that fluid, essentially. When we look at the cochlea, we've got our oval window up here at the top. So that's where the stapes would be attached. It's going to allow sound waves to enter into the cochlea, and those sound waves travel through this spiral to the end of the spiral, and then they basically reverse direction and go back out, and then are dissipated out that round window down at the bottom. So within the cochlea, if we look over here in this image to the right, this cutaway, you can see these two distinct tubes that are found within there. So we've got one that's kind of on the top and one that's kind of on the bottom. And again, the sound waves do travel initially through just one of those two tubes. When they reach the end of the line, then they essentially reverse and spiral back out through the other of those two tubes. So that's how we allow sound waves to enter the inner ear to the cochlea, and then how those sound waves then exit. So we enter the oval window and we exit from the round window. And again, it's filled with this kind of gelatinousy liquid that those pressure waves from the eardrum that have been transmitted through the bones then cause movement of this fluid inside of those chambers. Over on the other side, we've got a couple little structures here at the kind of at the base of the three loops. This is called the vestibule. And so a vestibule is like an entryway kind of. And so the vestibule for the inner ear function is kind of the first part that we kind of make sense of. And we've got two distinct structures within this vestibule. We have what's called the saccule and we have the utricle. And we'll talk about these more next week when we get into the chapter. For the quiz, you really just need to know the location of these structures. There's nothing that's asking you necessarily about function other than maybe the hearing part versus the balance and equilibrium part. But the saccule and the utricle help this part of the ear interpret essentially vertical and horizontal movement. So if you're, let's say I'm sitting at a stoplight and the light turns green and I punch it and I accelerate really quickly and my head pushes back like that. The utricle in particular is the part that senses horizontal motion. That quick movement of my head as that car accelerates, or let's say I slam on the brake and I decelerate, that's horizontal movement. That's going to cause that gelatinous fluid to move in that area. And where you see these kind of more like cell bodies, what's called the ampulla in here, I believe they're called the macula in the vestibule, that's really the sensory part. So the gelatinous part's all surrounding that, but the actual receptors that pick up that movement of the fluid are in these darker kind of purpley oval areas. The saccule is more about vertical motion. So if you get on an elevator and you're on the 10th floor and you push one, sometimes you feel like the elevator drops a little bit. The saccule is what's going to kind of pick that up. If you are on one of those amusement park rides that like brings you out and then like drops you all of a sudden, the saccule is going to be the part that really picks that up most discreetly. The three loops that we see, we've got an anterior, we've got a posterior, and a lateral. These are more about rotational motion, not necessarily planar motion or linear motion. These are more about angular motion. And so these are oriented basically to the three planes: sagittal plane orientation, transverse plane orientation, or frontal plane orientation. So when we look at the anterior, that's basically running front to back this way. So if I do something like a somersault, that's going to cause fluid to move in this tube, and that's going to trigger motion at these ampullas to let me know that, oh, I'm doing a somersault. I'm moving in the sagittal plane, but in a rotational function. When we look at the posterior, I believe it's the posterior is the one that is more about frontal plane motion. So if I'm doing like a cartwheel and I'm spinning that way, that's going to pick up that type of motion. And then that lateral plane is more in that transverse plane. So if I'm like, spinning around like that, that's going to be more of that particular one. So each of those three, what we call semicircular canals, senses rotational motion in a particular plane. So they have to work kind of cooperatively together to kind of help us figure out where our head is in space. And if you think about a sport like a gymnast or a diver, where they're flipping in multi-planar motions, they're doing a double backflip with a twist. Well, they're kind of going in all those different directions. And so they need to really be able to interpret accurately where I am in space, so they know and match it up with what are my eyes seeing. Even though I'm flying around and spinning at all these angles, my eyes are still giving me information too. So they kind of have to all be functioning together so that I know how to stick that landing or get that nice clean entry into the water because I went in really vertically, or any of those type of skills. So these are super important for high level skills, but they definitely play a big role with lower level skills, just walking and keeping our balance, trying to walk in a straight line and turn our head and talk to somebody at the same time, or keep our sense of direction when we're going up or down stairs, or starting and stopping. If you think about playing a sport and I'm sprinting up and then I have to stop and plant and cut, I really need all this information or else I'm not able to keep my balance. And so these are going to coordinate, again, with the cerebellum really closely, because the cerebellum has to help the muscles react to wherever we are in space so that we can stay upright. But this particular part of the brain is what really gives us that information about where our head is in space, and how quickly we're spinning in a given direction, or how quickly we're accelerating or decelerating, either vertically or horizontally. So they all have different features, and we'll get more into that a little bit later. But
so do you think if someone were having the vestibular testing done and they were having the nystagmus response where the eyes are shaking, but they couldn't feel it, they had no awareness of it, that's a cerebral issue, or maybe the information isn't being interpreted correctly?
The eyes, we do coordinate with the eyes, because the eyes kind of have to know where they need to focus based on where my head is in space. The most common reason why we have those sort of functions is that, we'll talk about it in a little bit, what helps us pick up that sensation. We have little calcium crystals that are inside of that gelatinous fluid that give it a little bit extra mass. And sometimes those calcium crystals, which should stay in the vestibule, sometimes they leak out and they get into these semicircular canals. And when they get into the semicircular canals, then they essentially make that gel be heavier, and it gives the brain the impression that there is movement in one of these planes taking place, even when there's not. And so the eyes start kind of freaking out. They're like, I'm getting a message that says I'm spinning around like this, but the eyes are like, I don't think I am moving that way. And so they just start trying to freak out, trying to make sense of it a little bit. So, we haven't talked about nystagmus, but and we will in some later coursework, but nystagmus is basically an involuntary twitching movement of the eyes, usually kind of side to side. And usually you can use that information to determine which of these canals might be messed up. And we can do techniques to try and help those calcium crystals find their way back home so that we can make those symptoms stop. And so that is something as people working in PT, we can do that technique on patients, and it can make a dramatic difference in fixing that problem, fixing that vestibular problem. Because it's awful. When people have really bad, what we call BPPV, benign paroxysmal positional vertigo, their whole world is spinning around and they can't do anything about it. They'll tend to be nauseous, throwing up, headaches. They'll feel just awful. And if you can correct it with this, the technique literally takes like 2 minutes, and you can often fix the person in one treatment, and they'll be like, it's a miracle. I don't feel bad anymore. But it can go wrong too, and you can make the person throw up and make them feel worse if you don't do it right. So it's not something you do lightly or go, I think this is how I do it. You definitely you have to know and be very sure of which way you have the position of their head turned to let that crystal move the right way.
Hey, Craig.
Yep.
Is that leakage also what causes the ringing in the ears?
I think the ringing, that would be much more likely to be dealing with the cochlea, because you're getting a sound, that buzzing sound in your ears. So that's some sort of defect going on with the cochlea. And a lot of times it's related to exposure to a lot of noise, things like that. So it's going to be not necessarily on the vestibular portion, but more on the cochlear portion that's causing some sort of defect with that.
So what about that sensation where you almost like hear silence? Do you know what I'm talking about when like silence is really loud that you can almost hear it?
I don't know if it's just that your cochlear nerve is getting a little bit more hypersensitive when you're in a quiet environment. I don't know that I've necessarily heard, maybe it's that it's an absence of noise or an absence of external stimulus that, I don't know, maybe you have like a background baseline level of activity in those receptors that for some reason when there's no fluid movement in there that it freaks them out in some way. I'm not 100% sure on that one. All right, let's look at some other structures. I don't think you need to know a lot about the makeup of the fluids, but you do definitely want to know where these three structures are: the vestibule, the semicircular canals, and the cochlea. Within the vestibule, you kind of want to know where the saccule is and where the utricle is. And then, yeah, we don't need to know that part too much. So there's our macula. The macula is basically where the receptors are for the vestibular portions, for the saccule and the utricle. So the macula is kind of where the sensory receptors are. The rest of the space within the utricle or the saccule is going to be that gelatinous fluid. And so that gelatinous fluid will move and it will trigger certain structures in the macula. Let me pull up to this slide right here. So this one is showing you kind of one individual portion of the macula. It kind of reminds me of a pair of hands holding it up. And the hair, these are called hair cells up at the top. It reminds me of the World, I think it's the World Series trophy that looks that way, kind of this angular thing. Anyways, they just won the World Series title. So we've got these different little receptors that are embedded and located in both the utricle and the saccule. They're going to work basically the same way. You've got this gelatinous fluid, and on all these little specks that you see in there are the otoliths. So the otoliths, sometimes you'll hear them called otoconia. Those are basically the calcium crystals. So they are quite literally little rocks in your head, very little. And they just give that gel extra mass. And so when you have that extra mass and that gel starts moving in response to acceleration, deceleration, dropping motions in the vertical or horizontal components, it's going to cause that gel to shift. The extra mass of the otoconia combined with that gel will make these hair cells bend. Imagine like seaweed flowing with water moving over it. That's kind of what's going on. When those hair cells move and change position, that's going to create an action potential from that receptor and send it down that nerve so that it goes to the part of the brain that needs to make sense of it. So those vestibular neurons that are kind of in that brain stem that we talked about during the lecture, that's where that message gets sent initially to help us understand, oh, you're accelerating, oh, you're decelerating, oh, you're falling down. So the shift of this movement causes a shift in these cilia. The kinocilium is the biggest, tallest one, like the king cilia at the top, and that's kind of the one that's the hardest to get to deflect. So when it's a bigger motion, then that's the one that's going to really start sending its signal to let us know this change in position or change in velocity of our body that we're sensing. And really the inner ear structures that help with that rotational motion, those semicircular canals, they're basically set up the same way. Instead of the macule, we have that ampulla part, but it's essentially the same thing. When the fluid starts moving, it makes those hair cells change position a little bit, and by bending and changing those little wires, essentially, that's what activates the nerve and sends that action potential so that we can pass that message on to the brain structures that need to interpret what's happening with that. All right, I think that's, yeah, kind of mostly what we need to talk about structure-wise. Again, within those semicircular canals, that should normally just have fluid. And sometimes, like I said, those otoconia can move past a membrane and get into those semicircular canals, and that's what can cause those abnormal signals that will often lead to vertigo, especially vertigo that comes on very suddenly, that doesn't necessarily have another cause. Sometimes a hard hit on the head can do it. It can cause those things to shift. For a lot of people, laying in a certain position might cause those things to move more. And we see those vertigo problems more with older individuals. The consistency of that gelatinous fluid is believed to kind of change a little bit as we age. And so that makes older people a little bit more susceptible to having these BPPV problems. But we will see vertigo problems with things like concussion injuries too, where that hard hit on the head can dislodge some of those little crystals and get them into the semicircular canals where they shouldn't be. They should stay in the vestibule area or in the ampulla area of the semicircular canals. So if we go back to this structure, actually, let me go back a little bit more. So again, we've got our semicircular canals here. Just where that ampulla is where these hair cells are going to be located. And the gel that's in the canal is going to move past those ampulla and cause that response to let us know, am I spinning this way? Am I spinning this way? Am I spinning this way? And kind of make sense of it, or some combination of all of those different motions. So the ear is unique in that it's got kind of that dual function. Most of our other sensory organs really just have one. We're either going to taste something, we're going to smell something, we're going to see something, we're going to feel something. I guess our sense of touch is kind of the other one that's a little different. The ear and the eye are considered to be sense organs. So it's not just a receptor. The receptor is part of a bigger structural system. And so, when we look at the eye, we have those anterior portions, the posterior portions, we have the retinal portions, we have the nerve portion, we have the rods, we have the cones, we have the cornea. If any of those parts are defective, it's going to ultimately affect the function of the eye. But with the ear, you can have distinct problems with just the cochlea that can, the vestibule, the utricle, stachyle, the canals, those might all be working just fine, and you might have hearing problems. Or you might have balance problems, but be able to hear just fine. So they do have, it's a shared common nerve, but it is distinct nerve branches that's carrying that information. So you can kind of distinctly separate those things out. When we look at our sense of taste, our sense of touch, our sense of smell, those are all more about kind of distinct individual receptors. They're not part of a bigger organ per se. So they work a little bit differently. And we'll get into all those, I believe, on Thursday of next week. So we've got about 20 minutes left, roughly, in class. So I think, do we have other questions on ear stuff? I didn't hear you. Before I was a PT, I worked at a clinic. We did fitness testing. And in addition to fitness testing, we would do a hearing screening and a vision screening and a few other things. And I can't tell you how many times I heard this joke. We would tell people, okay, we're going to do your hearing test. And the most common response we would get would be, huh? What? It's funny, like the first time you heard it, but when it was like the third time of the day you heard it, and the 20th time of the week you heard it, and the 1000th time of the month that you heard it, you kind of lost its humor. But, yeah. So don't try that joke on me. I've heard it way too many times. All right. So I think what we'll just do for the next 15, 20 minutes, I'll go ahead and just do your breakout rooms and let you guys kind of work through the labeling handouts for these eye and ear structures, and just kind of help you kind of reinforce some of that information so that you'll be a little bit more ready for the quiz. But again, don't worry so much. I don't believe there's any questions that ask about like functional stuff on the quiz. It's really all just structural. So understanding where the cornea is, understanding where the anterior chamber is versus the posterior chamber versus the anterior cavity and the posterior cavity, the rods versus the cones, the macula versus the fovea centralis, the sclera, all of those structural things you definitely need to know, and then all the ear stuff that we just talked about here. All right, let me, we don't have a ton of time, but we'll go ahead and I'm going to stop sharing, and I'm going to put you guys in breakout rooms real quick. And you'll just want to go into the lab activities, look for the labeling handouts. I think it's lab 10 probably is this week's. And so have at least one person in the group kind of pull that up and just get as far as you can on that. If you want to collectively work on the eye today and then worry about the ear on your own, or vice versa, you probably only have time to work on one of the two structures. But here we go. right, we got just about everybody back. Might have one or two people still to come. So anything that came up, any groups that I didn't get to, things you weren't clear on, based on what you're trying to go over with those eye and ear structures? All right. So, yeah.
Wait. My study buddy found something to help study cranial nerves, like something from the internet.
Uh-huh.
And it's like Olympic Olympians occupies troubled triathletes after finishing Vegas gambling vacations still high.
Okay. Instead of Harry Potter, that works for you.
And then it goes like Olympic that olfactory, Olympian opium. That's optic nerve, and it just along with it.
Yeah, whichever, any of those, if they help you keep them straight, then use whatever works for you, whatever helps it stick. That cranial nerve, we'll revisit that in a couple other classes. We'll spend some time on it in physiology next semester. But yeah, for the written exam coming up, you definitely want to know which nerve does what, which ones are sensory, which ones are motor, and which ones are both. I haven't really seen a good other little mnemonic saying for the functions besides the Some Say Merry Money one. But the one for the cranial nerve names, there's definitely a lot of different ones out there. So since many of you are from the Harry Potter generation, I like that one because it kind of sticks a little bit. I used to go to the book release parties with my son. I don't do well staying up late, but they'd be like the midnight ones. So I'd like have to take a nap so I could make it to the midnight release parties at the bookstores for those back in the old days. My son's 20, he'll be 24 in a couple weeks. So similar age to a lot of you guys probably. All right. Well, if we don't have other questions, I will see everybody on Tuesday. But
fun story. My sister got jealous of Harry Potter because it was taking all of our mom's time.
Yeah, we would kind of fight over the books.
Yeah.
You know, when we get the book, who got to read it? Oh, they put the book down. You went to bed. I can read 100 pages now. So yeah. All right. Y'all take care and let me know if you have other questions. Otherwise, I'll see you Tuesday.
Bye, everyone.
Quiz. Bye, Eric. Is our quiz on Tuesday?
Yeah.
Okay. For some reason, I was thinking it was a different day, but okay, good.
I think at some point there might be some that switched to Thursday, but I know I used to, but I think for this summer, I thought I set them all for Tuesdays.
Okay. Awesome. Thank you.
Yep.
Have a good weekend.
Thanks, too.
Craig, I have a question.
Okay.
So the three bones, the incus, the malleus, and the stapes, are they connected to each other with cartilage or something?
You know, I'm not 100%. I know they're not like synovial joints. I know you can get arthritis in them, so you can have some problems with movement between those joints where they'll get kind of frozen up a little bit, and that can lead to some hearing issues, some deafness issues if they can't transmit the sound waves hitting the tympanic membrane. But I don't know. I would guess they're probably, I mean, they have to move, so they can't be necessarily like fibrous where they're locked together, but I don't know exactly how they're linked.
Okay.
In collagen, I'm sure, but beyond that, I don't know.
What's the material for our quiz on Tuesday?
The eye and the ear.
Perfect.
Kind of all those eye and ear structures.
Thank you.
Yep.